What happens in the brain after losing a limb? The fate of the brain's sensory maps after an arm amputation has fascinated neuroscientists, clinicians, and anyone coping with limb loss. Are the neural representations for the missing hand preserved, change, or disappear altogether? The truth turns out to be far more nuanced than many realize, and stirs controversy among experts. Let's dive in. Our investigation centered on following individuals over time, before and after arm amputation, to uncover how their brain's organization changed. Why did we choose this approach? Most earlier research used cross-sectional designs, comparing different people in different states, some with amputations, some without. But cross-sectional comparisons can be misleading, thanks to natural differences among individuals. With small groups, it's especially hard to separate real effects from random noise. Instead, longitudinal studies track the same individuals through time, providing a unique window into how true reorganization unfolds. But here's the catch. Even longitudinal methods have limits. The brain can change for many unrelated reasons, like new MRI scanner models, or even participants becoming more comfortable with the testing environment, which could shift brain activity in subtle ways. To minimize these sources of error, we scanned both our amputee participants and a control group of healthy individuals across similarly spaced time points. For 2 amputees, we managed to follow up years after their amputation, although we were unable to collect matching data from the control group during those longer intervals. For group comparisons, 6-month post-amputation scans were the key reference point. Who participated? Across 7 years and numerous UK hospitals, we identified 18 candidates preparing for hand amputation. Strict criteria narrowed this field dramatically. Only 6 volunteers were safe and eligible for pre-surgery testing. Ultimately, just 3 completed all stages of evaluation. What made the process so selective? MRI safety, sufficient hand movement, appropriate age, and manageable levels of disability. All these factors determined inclusion. How did these surgeries differ? Each case was unique. Participant P1 faced a rapidly progressing blood vessel abnormality in the upper arm, requiring advanced surgical techniques. Besides simply cutting nerves, surgeons used targeted muscle reinnervation, redirecting residual nerves to new muscles and regenerative peripheral nerve interfaces, implanting nerve grafts, innovations offering better function, and perhaps enhancing future prosthetics. P2's amputation was much more conventional, prompted by a slow-growing arm tumor, with previous surgeries already limiting finger movement. P3's story involved Severell-Martorell syndrome and repeated bone fractures, resulting in another traditional amputation where nerves were left to retract naturally. The diversity in conditions and treatments, from pioneering approaches to classic methods, strengthens the credibility of our findings. Our control group consisted of 16 healthy older adults, average age 53, mostly right-handed, matched to the patient group in age and scan schedule. 4 additional controls dropped out before complete testing. All procedures received ethical approval, and every participant gave written informed consent for participation, storage, and sharing of their data. Digging deeper, supplemental datasets. For comparison, we tapped into prior studies, pooling 2 cross-sectional fMRI databases. Chronic amputees, 26 participants, average age 51, mostly missing left arms, with amputations ranging from wrist, forearm to above the elbow, and an average of 23 years since surgery. And another set of healthy controls. P1, being notably younger than the main control group, was matched with data from 22 young adults scanned twice a week apart. Assessments and questionnaires. Time windows for testing before amputation were often tight, with participants under significant distress and discomfort. This meant we prioritized neuroimaging results but still gathered valuable questionnaire data and functional ecological tests. Kinesthetic vividness. Participants rated how real finger movement felt individually before and after surgery. From 0, no sensation, to 100, as vivid as the other hand's fingers. Difficulty with finger movement. Participants indicated how hard it was to move each finger before and after amputation, ranging from 100, as easy as their healthy hand, to 0, most difficult imaginable. Pain reporting. Before and after surgery, participants rated both frequency and intensity of limb or phantom pain, using both standard and adapted questionnaires. Ratings covered pain over the past year and the worst pain in a typical week, along with nuanced descriptions using a large scale to capture subtle differences. Chronic pain was calculated by combining intensity and frequency, more accurately reflecting day-to-day suffering. We also measured vividness of painless phantom sensation and stump pain, and administered the Pain Detect and McGill Pain Questionnaire for comprehensive detail. Functional Index, using the Upper Extremity Functional Index. Participants scored their ability to perform various activities before and after amputation, quantifying impact on daily life. Ecological tasks. To explore real-world adaptations, some participants performed a present wrapping task, designed to reveal habitual compensation strategies. Their efforts were video recorded. Finger movement and kinematic tests. We asked participants to perform isolated finger movements on cue during each session, reporting their performance, including attempts with phantom fingers. We also invited 2 amputees for special sessions assessing whether phantom movements involved covert activity in the intact hand. MRI scanning procedures and fMRI task design for each scan. We obtained structural images, 4 finger mapping fMRI scans, and 2 body localizer scans. Cross-sectional datasets followed similar protocols. Finger mapping. Our main task instructed participants to make specific movements, cued visually. Individual finger movement, toe curling, lip pursing or resting across 13 conditions. For amputees, we adapted methods to simulate phantom sensation, elevating the affected hand so movements were performed in the air. The control group matched these procedures, half using the right, half using the left hand. Cues were presented as flashing vertical bars representing each finger, or with words like feet or lips. Each movement condition lasted 9 seconds, repeated 4 times per scan, with rest periods before and after. Imagery controls. In additional scans, participants were prompted to actually move or imagine moving various body parts, adding further comparison conditions. Each was presented in 10-second blocks, repeated throughout the scan. Technical details. MRI scans used a 3T Siemens Prisma with a 32-channel head coil, collecting both anatomical and functional data, with specific high-resolution settings to ensure detailed cortical coverage. Every scan included checks for motion and noise, with problematic scans flagged and corrected. Data analysis and pre-processing. Data were processed using recognized neuroimaging software, FSL, FreeSurfer, MATLAB, Python, carefully correcting for motion, smoothing, and signal normalization. To ensure all longitudinal scans were aligned for each participant, we computed a mid-space average for structural images, mapping all functional data to this common space. For statistical modeling, we applied general linear models, GLM, to extract activity contrasts, movement versus rest, or among different fingers' body parts. Selectivity was analyzed by contrasting each finger against